Hello and welcome back to another Mr. Carter Science special video. This particular video will be following the Edexcel specifications for biology and double science, and we'll be looking largely at digestive enzymes. As always, please pay attention and make certain you follow the specification in your curriculum links. Make certain that you pause this video often, make notes and try and answer the questions that come along in the video. So you may have seen this particular experiment in school. It's a nice way that teachers often use to introduce the concepts of enzymes, and it's called the model gut experiment. And we start by having a test tube of water with a very special kind of plastic bag called a Viscin tubing, and inside the Viscin tubing there is some starch and some amylase. As we go through this PowerPoint, I will try my best to underline the ending of any enzyme names, because for most enzymes end in the word "ase." Helps you to identify which chemicals are enzymes. So this is an experiment used to sort of introduce the concept of what's going on. At the beginning, we've got starch and amylase in our Viscin tubing and nothing in the test tube apart from water. After 5 minutes, the teacher will probably stop and ask you to test the contents of the Viscin tubing and of the water. And what you'll find is after 5 minutes, that's the wrong colour. Let's try that again. So after 5 minutes, inside the Viscin tubing, you will have no starch. You might use the iodine test and you'll find that there is no starch inside the Viscin tubing, but we have proteins present in the form of the enzyme amylase, and that we have sugars present. So you may have used a Benedict solution. So the question is, what's happened to all of that starch? Well, it's clearly been broken down and turned into sugars. But after 5 minutes in the test tube, we also have no starch, but there are no proteins, but we do have sugars. So this proves to you a couple of things. Firstly, that proteins are large molecules and they cannot pass through the holes in the Viscin tubing. Sugars are small molecules and they can pass through the holes in the Viscin tubing. This is why it's used to model the small intestine, to show to you that small molecules can move through the linings of the small intestine, and in this case the Viscin tubing. And it also proves to you that this enzyme amylase will turn starch into sugars. So what is an enzyme? Well, enzymes are proteins. They are biological catalysts that speed up chemical reactions. So we've got a classic example of an enzyme here, an enzyme diagram, and we have this special area here which is known as the active site. So the whole green area is our enzyme, and that molecule is made out of proteins, and like any protein, it's made out of smaller amino acids all joined together to form a big protein. So enzymes are biological catalysts. And any catalyst, whether it's in biology or in chemistry, is a chemical that will speed up the chemical reaction, the rate of reaction. But catalysts are not used up in the reaction themselves. So at the start of the reaction, however many grams of your catalyst you have, you'll have the same amount at the end of your reaction and you can reuse them. So how exactly do these enzymes speed up these chemical reactions? Well, they do that by lowering the activation energy. So if we have a little look at this graph down here, you'll see that without the enzyme present, our reactants need to have added this much energy before the chemical reaction will start. If we have an enzyme present, we only need to add this much energy. There is a difference this much on this particular graph. So that means in terms of your body, in terms of cells and your temperature, which is 37 degrees inside your body, you can have the same chemical reactions happening but at a much lower temperature. So this slide is explaining to you the classic lock and key mechanism of an enzyme, whereby we have our substrate here, and a substrate is any chemical which will fit into the enzyme's active site. Now you will only get one specific substrate which can fit the specific shape of the enzyme's active site, and they will form together a complex. It's actually known as an enzyme substrate complex. And a chemical reaction will take place and the substrate will be turned into our products. So in terms of the equation, you would say the enzyme plus the substrate forms an enzyme substrate complex, and then we have the enzyme left over ready to react again and our products have been made. So there's three main groups of enzymes that you need to know about. We have the first group here, which are the carbohydrases. Carbohydrases. Again, underlining the A's. There's a couple of different enzymes in your body that fit into this carbohydrase group, and these are enzymes that break down carbohydrates. For example, we have the enzyme amylase, which will break down starches into sugars, in particular the sugar maltose, and this occurs in the mouth and the small intestine. This particular enzyme is produced in the salivary gland, in the mouth, and also in the pancreas, and it's found in the small intestine. So here we have our large starch molecule, and we have the amylase here, and that amylase is breaking up this large insoluble molecule into our smaller soluble sugar maltose. But then we have a second enzyme, maltase, and the maltase breaks up the maltose and turns it into smaller molecules called glucose. So maltase is found in the lining of the small intestine, in the lining of the villi and the epithelial cells. So we can have two types of enzymes working here. As is clear for almost all enzymes in your body, they're trying to break down large insoluble molecules into small soluble molecules. And that's also true here with the protease enzymes. Protease enzymes break down proteins into amino acids. Proteins are large insoluble molecules. Amino acids are small soluble molecules. So here's our particular protease enzyme breaking down proteins into small amino acids. Protease enzymes, let's underline the A's again, are quite a large group of different enzymes. In fact, there's many different protease enzymes. Some that you may come across, you might come across the enzyme trypsin or the enzyme pectin. Both of these are protease enzymes and they break down proteins. The last group of enzymes we need to look at are the lipase enzymes. And lipase breaks down fat and oils into fatty acids and glycerol in the small intestine. So here is a molecule of fat made out of our two different molecules, glycerol and fatty acids. And the enzyme lipase will break these bonds here that hold them together, forming our glycerol and three fatty acid molecules. So lipase is produced in the pancreas and found in the small intestine. Let's have a quick task to see how much you've learned from the first few slides. Pause the video now, draw down this table and see if you can fill in the missing gaps. Remember, you're trying to identify the name of the enzyme, the substrates that they break up, the products that are made, and we'd like to know the organs where the enzyme is found. So pause the video now. And here are the answers. Tables like this are very useful because they contain a large amount of information but without a large amount of writing. So you might find it much easier to produce a table in your revision and then be able to reproduce this in full sentences in your exams. I'll give you an example. The enzyme amylase is used to break down starch into maltose, and amylase is found in the mouth and small intestine and pancreas. We can do the same thing for each one. Maltase breaks down the carbohydrate maltose into glucose. Maltase is found in the small intestine and the epithelial cells of the villi. Proteases, like trypsin and pectin, break down proteins into amino acids, and protease enzymes are found in the stomach and the small intestine. At this point, it's very important to say that the only group of enzymes we find in the stomach are proteases. So the only chemical that gets broken down and digested in the stomach is protein. No other chemicals are digested in the stomach. The enzyme lipase breaks down fats and lipids into glycerol and fatty acid, and lipase is produced in the pancreas and the small intestine. In this particular example, it's very important to know that we have two names for the word fat. Fat can be known as fats, or they can be known by the slightly more accurate chemical name of lipids, and that's why the enzyme is called lipase, because lipase breaks down lipids, which are also fats. The final enzyme in our group here is one that's often used in biology lessons because it's very easy to find. We can find it in just a little piece of liver or in some potatoes. And that final enzyme is called catalase. And catalase will break down hydrogen peroxide, which is H2O2, into water, H2O, and oxygen, O2. So you may well have done a large number of experiments using catalase to try and work out the rate of a reaction. And that's what we're going to look at next. But before we do look at rates of reaction, please try these enzyme review questions. Again, pause the video, write the questions and answers down before looking at the answers on the next slide. So here are the answers. Define the term enzyme. This is often a 3-mark or a 2-mark question in an exam. So you need to say, firstly, that enzymes are proteins, they are biological catalysts, and then you need to explain that by saying that they speed up chemical reactions. What 3 letters do most enzymes end in? Most enzymes end in A's. Lipase, amylase, maltase, catalase, protease. A couple of exceptions would be trypsin and pectin. Explain what digestive enzymes do. Well, digestive enzymes in general, they will break large, insoluble molecules into small, soluble molecules. If we want to be more specific, we've got questions 4, 5, 6 and 7. So describe a function of amylase. Amylase breaks down starch into small sugars, which often gets you a mark, or more specifically, maltose. The function of maltase is to break down maltose into glucose. The function of protease is to break down proteins into amino acids. I often use the abbreviation A dot for amino acids. And lipases break down fats or lipids into glycerol and fatty acids. So the next thing we need to consider are rates of reaction with enzyme controlled reactions, and in particular, how does temperature and pH affect the rate of an enzyme controlled reaction. You may well have done an experiment very similar to this in the classroom, where you take a test tube of starch solution and you add to it some amylase. You make certain they're both exactly at the same temperature, and then every minute or every 2 minutes you remove a small amount of the solution and you put it into a spotting dish here. These are some spotting dishes or spotting trays, and each well of a spotting tray contains some iodine. And in that way you can tell whether or not starch is present. So a student who's done these results has done this experiment 3 times at 3 different temperatures. They've done it at the bottom at 20 degrees C, then here at 37 degrees C, and here at 80 degrees C. So if we just look at the results for 37 degrees at the moment, which is this line here, we can see that at time 0, the student has carefully written the times down here: 0, 2 minutes, 4 minutes, 6, 8, 10, 12, and finally 14 minutes. At time 0, the iodine has turned blue-black, and that tells you that there is starch present. At 2 minutes later, the iodine is a yellow-brown colour, which tells you that there is no starch present. At 37 degrees, all the starch has been broken down within the first 2 minutes of the experiment. But at 20 degrees, even at 4 minutes, we still have the blue-black colour. There's still starch present in the solution. It takes between 4 and 6 minutes for all of that starch to be broken down. Now, we don't know exactly how long. We can only say that by 6 minutes there was no starch left. So we know that it took between 4 and 6 minutes for all the starch to be broken down. At 80 degrees, we can see even at 14 minutes the iodine is still a blue-black colour. There is still starch present. None of the starch has been broken down even 14 minutes after the reaction has taken place. So this experiment tells you how much time it takes for amylase to break down all the starch in the solution. But we can also then use the results of these experiments to calculate a rate of reaction. And the rate of reaction is how much product, how much maltose in this case, is formed per minute. Per minute. Rates of reaction are always how much product per minute. So, looking at the results of this experiment, they may ask you questions very similar to these 3 in the exam. So pause the video for a minute, have a think about the answers, and I'll come back to you in a moment with the actual answers. So one safety precaution you should always use when carrying out these experiments is to wear goggles. Enzymes are irritants. They can break down the various chemicals in your eyes and on your skin, so you should always be very careful and wear gloves and goggles to protect yourself. In particular, also we're using iodine, and iodine can stain and damage the cells of your eye, so again goggles would be the main safety precaution that you would use when carrying out this experiment. Goggles. Name the independent variable. Well, as we said before, we've got 1, 2, 3 different temperatures. They have decided and chosen to change those temperatures, so the independent variable is the temperature. Explain how many minutes it took for a reaction to be completed at 20 degrees. Well, as we said previously, it took between 4 to 6 minutes. So you will generally get 1 mark for that in an exam, but you've got to be careful because it says explain. So we have to say, how do we know that? How do we know? Well, we know that because the blue-black colour had changed to yellow, and this means all the starch had been broken down. So if we take those results that we have from that experiment, we can then use those results to plot a rate of reaction. And you generally get a graph that looks like this. So when you have a graph that looks like this, there's a few key things you will need to be able to do. You must be able to describe the graph and explain the graph. So let's look at the first area where we're describing the graph. Whenever you have to describe a graph, you should always start the sentence with as the. And then you look at the bottom of the graph and you just state what's written at the bottom as the temperature, and then you include the word increases. As the temperature increases, these numbers here are all getting bigger. It's a very simple start. As the temperature increases, then we carry on our sentence to say the. As the temperature increases, the. And we look at the other axis, the y-axis, and all we do is copy it down. As the temperature increases, the rate of reaction, and then we have to think, and this is the only hard bit, what's happening to our line here? It's increasing. As the temperature increases, the rate of reaction increases up to this point here, 37 degrees. This is known as the optimum temperature. As the temperature increases, the rate of reaction increases up to an optimum temperature of 37 degrees. But we've only described half the graph. We then need to use our sentence again. After 37 degrees, as the temperature increases, the rate of the reaction decreases. And all we've done is describe. We haven't said why. But if your question asks you to describe, that's all it's asking for. As the temperature increases, the rate of reaction increases to an optimum temperature of 37 degrees. Then, as the temperature increases, the rate of reaction decreases. But we may well be asked to explain. So why does this happen? Well, the first thing we need to think about is the first section of the graph down here. As the temperature is increasing, the rate of reaction is increasing, and this is because those molecules are gaining heat energy, and that means that they're moving faster, so they're gaining kinetic energy, and that means that there's going to be more collisions between the enzyme's active site and the substrate. Remember, it has to be the enzyme's active site. It's no good if the substrate hits the wrong side of the enzyme. It's got to hit into the active site. So as the temperature increases, there's more kinetic energy, more successful collisions between the enzyme's active site and the substrate. But after 37 degrees, the rate of reaction begins to decrease. And this is because the enzymes begin to denature. And denaturing is when all the bonds that hold together the enzyme in that very specific shape and hold its active site in the specific shape, they begin to break down. So the active site changes shape, so the enzyme can no longer catalyze the chemical reaction, so the rate of reaction decreases. So what does that denaturing look like? Well, if this is our normal enzyme's active site here, over here, the active site has changed shape. It's been denatured. The substrate can no longer fit into that active site. And enzymes will be denatured at high temperatures, generally above 40 degrees. And they will also be denatured by changes in the pH. All enzymes will have a particular optimum pH, but if you move away from that pH, either lower or higher, then the enzyme's active site will denature. So let's see. Have a look at this graph, pause the video, copy down these two paragraphs and fill in the gaps. Can you describe and explain this graph? What happens when we increase the temperature and we measure the rates of reaction for an enzyme controlled reaction? So here are the answers. As the temperature increases, the rate of reaction increases up to an optimum temperature of 37 degrees. After this, as the temperature increases, the rate of reaction decreases. We have successfully described the graph, but we haven't explained it. Explaining, generally you might get up to 6 marks for a question like this because we have to say why. Anytime you see this word, it means say why. Why does this happen? So, as the temperature increases, the particles gain kinetic energy, the particles move faster, there are more successful collisions between the substrate and the enzyme's active site, so the rate of reaction increases up to an optimum temperature of 37 degrees. After this temperature, the enzyme's active site changes shape and becomes denatured. The substrate no longer fits in the active site, so the rate of reaction will decrease. Did you get all of those correct? If you didn't, please go back, make your corrections, rewrite these two paragraphs to make certain you can describe and explain this graph. What about pH and enzymes? Well, again, we've got a very similar graph here, but in terms of changing temperature, we have now increased the pH and measured the rate of reaction for an enzyme controlled reaction. What I'd like you to do is to look at the next slide, pause the video, copy out the two paragraphs. Can you describe and explain what's happening in this graph? So here are the answers. First of all, let's try and describe the graph. As the pH increases, the rate of reaction increases up to an optimum pH of 8. After this, as the pH increases, the rate of reaction decreases. Can we explain why? As the pH increases, the enzyme's active sites change shape and fit the substrate better. So there are more successful collisions between the substrate and the enzyme's active site. So the rate of reaction increases up to an optimum pH of 8. After this pH, the enzyme's active site changes shape, becomes denatured, and the substrate no longer fits into the active site, so the rate of reaction decreases. If you can remember these paragraphs, you should be able to pick up a large amount of marks in your exam. You just have to understand when to apply this knowledge to what particular questions. Thank you very much for watching this video on enzymes and digestion. I hope that you've learned some things. Perhaps you can comment below with your learning and any improvements you would like from these videos. Please don't forget to like and subscribe. Thank you very much for watching this Mr. Carter Science Special.